completely identical. There is some variation. The variation appears as a result of mutation. Like, for example, here we can have a bug that is green, and then there's a mutation in a gene, and the bug now is brown. Okay, so the idea is that there are biological mechanisms like mutation that generate diversity in a population. Now, once we have a diverse population, it can happen that organisms with different traits are differently adapted for survival. Some of them may have faster replication. Some other may be able to escape predators because they mimic with the environment. And as a result of that, what we have is that in this heterogeneous population, some individuals are going to have higher chances of survival or reproduction than others. Like here in the example, if being brown helps you escape from the predator, then after some time, we will have that most of the population is brown. So what we have seen is an evolutionary process from a population that was green to a population that is brown. And this second ingredient in evolution is called natural selection. Now, this is not all, because in nature there are also, random events. You may just have bad luck. You may have this population of bugs, and then someone not very carefully walks over them, and you know it can happen. Like just steps on top of the 3 green bugs, and now all the population is brown. And this has nothing to do with being more fit or like being more able to survive. It's just bad luck. Of course, if we have 1,000 bugs, what's the probability that you step on all the green ones? That's very low. But if we only have, like in this example, 7, it's like throwing a dice. It can happen. So the effect of these random effects, which are called technically genetic drift, have to do with the population size. When we have very large populations, the mechanism that dominates is natural selection. When the population is small, this random drift becomes dominant. This is an important point. I will come over this later in the presentation. Now, these are the 3, let's say, major ingredients in modern evolutionary synthesis or in classical evolutionary theory. When we talk about microbes, we have to include some more ingredients that may not be exclusive of microbes or prokaryotes, but they are especially important when we think about those things, the microbial world. The first one is horizontal gene transfer, which is the following. It's the idea that bacteria and archaea can transfer genes from one to another through several mechanisms. So now we have that genes in the population, or the frequency of genes, does not only change because of natural selection or because of chance, but also because these genes are being actively transmitted, and these variants are actively transmitted between organisms, between individuals. And then we have a very intense arms race between parasites and hosts. In this case, parasites are normally viruses, bacteriophages, and archaeal viruses, and hosts are the bacteria and archaea. And of course, that also happens in the world of animals. Like there are predators and preys develop mechanisms to escape predators. But I would argue that that's even more, that pressure is even stronger in the case of microbes. There are estimates of how many bacteria are killed by bacteriophages in the sea, like every day, and the numbers, I don't remember the numbers out of the top of my head, but the idea is that viruses are one of the main mechanisms keeping bacterial populations stable in the sea. So, of course, bacteria have evolved mechanisms to escape viruses. Viruses have evolved mechanisms to escape the defense mechanism of the bacteria, and so on and so forth. So we have this never-ending arms race that affects the evolution of bacterial and viral genomes. So I'm going to present today probably 3 of these topics that have to do with evolution in the case of prokaryotes and how that evolution reflects in the composition of prokaryotic genomes. Also, I will give a disclaimer here. So far, I've been talking of microbes. I used the word microbes in the presentation, and I'm not being very rigorous because I'm going to talk about prokaryotes, bacteria and archaea. Okay, I want to make this disclaimer here. So the first big question, I'm talking about evolution, about adding microbes to the evolutionary theory. And we'll probably know that there is a paradigm of evolution. If there is a figure that represents what evolution means, if we have to just simplify that to a concept, we would use the concept of the tree of life. This is the famous drawing by Darwin. So the first realization of what an evolutionary tree could look like. So the first idea is, does this paradigm also apply to microbes, to to prokaryotes and viruses, or it doesn't. So to give you a bit of a historical background, the idea of an evolutionary tree was first proposed by Darwin. He just proposed it as something theoretical, like as a concept. The first person who actually tried to take real organisms and put them into a tree that describes the evolutionary relationship between them was Haeckel in the second half of the 19th century. And here there is one example of that tree made by Haeckel. If you look where prokaryotes are in that tree, there is only one single representative here in a red circle. It's not very visible due to resolution problems, but it says Vibrio, I think. Vibrio is a bacteria, as you know. So that was all. And it's normal that there was only one point and all of them were there first because there was not a lot of knowledge about microorganisms at that time, specifically bacteria. And second, because this tree is based on morphological traits. How similar are different organisms? And if you look at bacteria in the microscope, there is not much differences. Of course, there are differences in shape. The stain can be different, but it's difficult to say just based on that how they are related to each other. Things changed much later in 1990 when Carl Huss decided to build an evolutionary tree based on molecular comparison. So he looked at a single gene, a ribosomal RNA, and he used this gene as a marker to try to trace the evolutionary relationships. And say the main finding of this approach was to find that what we consider like a minor branch of the tree of life, prokaryotes, actually happen to constitute 2/3 of it here. And actually it splits in two groups or two domains, the archaea and the bacteria, which are as different within each other as they are with respect to eukaryotes. So this completely changed the picture. Now prokaryotes become the majority of the tree of life, and moreover, they are very different within each other. There are two groups that are completely different to each other. But that was only based on one single gene or on one single molecular marker. What happens if you look at different genes? Because of course, you know, a gene is just a tiny part of a genome. And then what happens is that things get more and more complicated because you compare different genes and the trees that you get are not consistent with each other. There is some incompatibility, and that's due to horizontal gene transfer. The idea that genes can be transmitted from one individual or from one species to another species. And if we go back at the base of this Doolittle called the thicket of life, this horizontal transfer occurred even between different domains of life. There is horizontal transfer between archaea and eukarya, archaea and bacteria, bacteria and eukarya. So things get tangled and difficult to grasp. And of course, the question is, well, with all this horizontal gene transfer, does it make sense to talk of a tree of life or it doesn't? If each gene has a different history, can we talk of a species level tree? Or we can only talk about gene level phylogenetic trees? This was a question that stayed around for a long time. I think it was quite convincingly closed just around 10 years ago by Per Puigbo from Gene Kunin's group. Basically, what he did was to take many genes, build the trees for each gene, try to put them together, see if there is overlap, and what he found was something like this. It's what they call it the forest of life. So you can see that there is a clear background of consistent trees, which would be basically what you can interpret as a species tree. And on top of that, there are like these like horizontal connections that have to do with horizontal transfer. So they actually quantified what's the percentage of the tree that, or what's the percentage of this graph that has a tree-like evolution, and what's the percentage of horizontal gene transfer. And they concluded that we can still talk of a tree of life in a statistical sense for prokaryotes. But what about viruses? Things here get far more complicated. First, they diverge very fast. So even if we look at single genes, we can only build trees for very narrow groups of viruses. Okay, we can take a gene and build a tree that is reliable for many viruses or for very diverse viruses. And there are no universal genes. We don't have a ribosomal RNA gene that we can use to at least have one gene tree. So things don't look very good. So we came up, we found this problem there, and we decided it was time to try to solve it. And to do that, what we propose is to use network analysis to overcome the limitations of these classical tree-based phylogenetic methods. So how does this work? We start with a collection of viral genomes, and for each viral genome here, we identify the genes that it has, and then we would connect the genome to the genes. Okay, so genome and genes. Now we do that for many different genomes. So far, just we have the genomes connected to the genes. The next step is to identify which genes corresponds to each other. So we have this virus here with a gene that does this function, another virus with a gene that does the same function. Both genes are very similar, both have the same origin. So we can say that, for example, these 3 genes are equivalent, these 4 genes are equivalent. And now we just take the genes that are equivalent and create a gene family. And what we get now is something that is called a bipartite network. Now you see this is a real network, but it has 2 kinds of nodes. On the one side, we have the viruses. On the other side, we have the viral gene families. And then we have a connection if a particular gene family is present in the genome of a particular virus. So this representation of the contents of viral genomes, bipartite network, contains all the information about the gene sharing patterns between viruses. And we can use that to try to reconstruct or to try to find relationships between viruses, just to show you how it looks like. Here in color, we have the viral genomes and the intersections between the edges, like here, this one, these ones here that have no colors and are tiny, represent the genes. Okay, and on top of that, we have clusterized the genomes based on communities of the network. So I will talk about this later, but this is more or less what things look like. It's very nice, and virologists like it a lot because it gives you a very nice visual impression of what the genomes of a virus contain. You can label the viruses based on their names or their taxonomy. You can also find these points that are highly connected, which are the genes that are shared by many different viral species, and you can label them so you can see what they are sharing with each other. But our point is that you can use it for far more than this kind of just visualization of genome contents. So how do you extract it? How do you extract more information from the gene sharing network? To do that, we can use some network analysis tools, such as community detection. So now I will introduce this concept of a modular network. Basically, as you can see here, a community or a module or a cluster in a network is a set of nodes that are densely connected to each other and weakly connected with nodes of other communities. If a network has that kind of a structure, like here, in this case, we have three modules, 3 communities that are, you see, they're densely connected with each other and almost no connection with the rest. So these kind of networks are called modular networks. And there are tools that allow identifying these communities, these modules within the network. Now, from a sort of evolutionary perspective, we can understand that viruses that share many genes probably also share common ancestry. I will try to convince you about this soon. So, module is a group of viruses with significant overlap in their gene content. It can be understood as a taxonomic unit, and it can have to do with the shared evolutionary history of those viruses. Here I'm showing you the example of the RNA virus network. So in this case, it's not a viral genome, but a community of the network, of the lower level network. And what we did was to compare the taxonomical assignments of each module in this network. And we found that there is a very good agreement, and almost one-to-one agreement between the modules of the network and the classical taxonomical classification. So that's telling us that this network approach can be used to classify viruses. If we just get a new virus that we have sequenced and we don't know where it comes from, we can just predict the genes, find homology between those genes and known gene families, put that in the network, see in which community it goes, and that can be used as a way to assign taxonomy in a more or less unsupervised way. So this approach, this framework is coherent with the supervised taxonomical classification of viruses. And then we can go further because this idea of modular structure can be applied in a hierarchical manner. So we can go up several levels. We start with first level modules like these 3 ones, but then these modules are also connected with each other and not with the rest. So we can have like second order modules, third order modules, and so on and so forth. So we get a hierarchy of modules. Why is that interesting? Because we can use that to generate sort of tree, I'm not going to call it a phylogenetic tree, of course. We can call it more like a dendrogram, in which we can actually trace these similarities in genome content at different levels. So this example is for all double-stranded DNA viruses. The network is here, again as before, not the genomes, but are the modules. And what we found doing this, using this approach, is that there are like two big groups of viruses. One would be represented by these modules, 3, 4, and 5, which are these ones here. And the other is represented by these other modules, which will be these ones here to the left. And it makes a lot of sense if you look at the molecular biology and the genomics of these viruses. All the viruses in this side, here on the right, this set here, have a specific set of proteins involved in morphogenesis, so involved in making the capsid and packaging the DNA into the capsid. And then the viruses here have a different set of morphogenetic proteins, different kind of capsid and different encapsulation proteins. And despite all the horizontal transfer that there is, we saw a lot of genes that are shared by members of the two large families or the two large supergroups. There is no event of horizontal gene transfer involving these morphogenetic proteins. So it's telling us something important about proteins that cannot be exchanged, that cannot be replaced, and that also have to do with the global structure of the virosphere. I will also point out that each of these two like supergroups contains viruses that infect bacteria, archaea, and eukaryotes, which is suggesting that this like diversification of double-stranded DNA viruses in two like giant groups happened before the diversification of the three domains of life. Okay. It's highly unlikely that a virus that infects bacteria changes enough to be able to infect eukaryotes once that it has specialized to infect bacteria or the same with archaea. So this will be the split. We can also use the network to identify what we call viral hallmark genes and niche-specific genes. The hallmark genes are these ones that are in the middle, the more central, that are shared by all the clusters from the same viral superfamily, while the niche-specific genes are these ones that are shared by groups of viruses that are taxonomically very diverse, but however share the same niche. This case here that I'm pointing out, those correspond to genes that are shared by viruses that infect mycobacteria, and those genes are required to break the cell wall of mycobacteria. So we can try to map these events of, we can try to identify these genes that have to do with niche adaptation by looking at the network and finding genes shared by modules that are very, very different in the network. We also did some research on the network of archaeal viruses, and we found something that was quite surprising at the time, although not completely unexpected. So here again, now I am representing viral genomes on modules, and the colors represent the modules. This will be just the archaeal viruses, and here we have the whole network, the archaeal viruses here, and the rest of double-stranded DNA viruses here. And I think it's quite shocking, like you can clearly see the difference in the density of connections. Like here, everything is connected with everything, like there is a lot of gene exchange. In the case of the archaeal, the network is much more disconnected. And then if we zoom into the network to the left and we look at those genes that are shared by, say the state, like this one, these genes that are shared by many different archaeal virus families, we saw that those genes generally, well, these ones have to do with the morphogenesis, but these ones here, especially the ones that are present in extremophile archaea, are genes that do not have much to do with viral function. Those are genes that are very widely distributed within the among the hosts that those bacteria infect. May have to do with living in like extreme environments, like highly acidic or high temperature environments. So they probably don't imply any kind of common ancestry. They just are this kind of niche-specific genes that I showed you before. What this is suggesting us is that unlike most of other double-stranded DNA viruses, archaeal viruses probably have appeared in different occasions during evolution, and these different families have independent origins. And also, there is something related to the biology of these viruses or the environments in which they live or their hosts that is preventing widespread horizontal gene transfer. So this is generating a series of hypotheses and questions that would be worth investigating in the future. this is all about, so what I'm going to talk about is virus gene sharing networks and how they can be used to overcome the limitations of the tree paradigm in the case of viruses. I'm going to go now with two more questions. So now we're going to move from viruses to bacteria and archaea, but we're still going to talk about genomes. The motivation for this research question has something to do with the recent changes in the way we are studying evolution in the last decades. So we go back to Darwin and Lamarck's times. Most of evolutionary studies, let's say many of evolutionary studies, were based on comparative anatomy and physiology. They look at the shape, the traits, the function of the organisms, and based on that, they develop evolutionary theory. And the main conclusion is that natural selection traits tends to improve those traits that enhance survival. So, to the point that natural selection is effective, we are going to find organisms that have traits that help them survive and make them somehow efficient. Now, we are in the area of comparative genomics. So it's very natural to ask ourselves if the genomes are also the result of selection. To what extent genomes are optimized to be more efficient in replicating or in providing the host with the traits that help the host surviving. This question can seem a bit tricky or trivial to you. You would say, of course, like the traits of an organism are codified or coded by the genome. So if evolution tends to promote fitter organisms, of course, that's going to imply that the genome is more optimal. But it's not necessarily like that. You may be familiar with this idea of the selfish gene. Selfish gene is a gene, a real genes, they have been described, that provide no clear benefit to the host. They don't have any function that seems to be useful for the survival of the host. They just seem to be selfish. They stay there. They are able to propagate by passing from generation to generation, stay in the genome. They are a little bit like genetic parasites. They are just taking advantage of the genome where they are to survive, but they are not contributing in any sense to the well-being of the host. And actually, this creates some sort of paradox, because if selection is acting to promote the fitness of the host, how is it possible that it allows for persistence of these selfish genes? Well, this is one of the questions that we want to answer with this research. So we know that not everything in a genome is adaptive. So genomes are not 100% streamlined and optimized to have only beneficial functions in them. Now I'm going to go back to this idea of how population size affects evolution. So this would be the naive expectation. In theory, when we talk about prokaryotes, by the way, bacteria and archaean bacteria especially have very large population sizes. Some exceptions like obligate intracellular endosymbionts, but in general they have very large population sizes. So we would be in this right column here of the table. Okay, large population size means that the selection is very efficient. Because of that, it's going to be able to keep genes that are only slightly beneficial. They are not very beneficial, but just a little bit. But because the population is very large, you still can see them as beneficial and keep them. As a result, you are going to be able to keep those genes, so the genome will be large. And in theory, we would expect that these selfish genes that are not beneficial for the organism should be eliminated. That's the theory. What happens in practice? Well, we see that there is actually a correlation between genome size and the efficacy of selection, which means that these two predictions hold. However, what happens if we look at the selfish genes? We see that they also correlate in abundance with genome size and in theory with strength of selection. So the more efficient selection is, apparently the more selfish genes you have. This is something weird. What's going on here? We'll see. It's also been found in experiments that selection can promote gene loss. So sometimes selection does not help you keep genes that could be beneficial, but it helps you remove those genes that are not being possibly useful at that particular time. And what that means is that genome evolution is a bit more complicated than this simple, simplistic picture I put or table I put in the top, because it's the result of an interplay between the adaptive value of a gene, the average adaptive value over time, and also, and we will see later, the rates of horizontal gene transfer, of gene gain and loss. So just measuring the effect of a gene in the lab in a given experiment is not giving us an idea of how important that gene is or how beneficial it is in the long term in evolutionary timescales. And that's basically what we wanted to do with this project, try to quantify the adaptive value of different functional classes of genes at longer evolutionary timescales, and use that to understand how selfish genes persist. It could be that they have some beneficial functions that haven't been described, or that they only manifest very occasionally, or it could be that there are other mechanisms involved in their persistence. To do that, well, let me go one step later. So these are the mechanisms that allow a gene to stay that govern genome dynamics. So on the one side, we have, let's start here with horizontal gene transfer. You can have a gene that arrives from outside and you didn't have it, and now you have it. There is duplication. So you have one copy and now you have two copies of the gene, and there is loss. It can be deletion, and then you lose a gene. And these are all neutral processes. There is no selection in here. There's just things that happen, things that generate diversity. On top of that, we have selection, which basically means that there is going to be a differential growth depending on how many copies of the gene you have. It can also involve depending on whether you have the gene or you don't have it. Okay, so that's the kind of process that govern this genome evolution. Now the problem is that when we look at a collection of genomes, we have the evolutionary tree, we have the composition, the presence or absence, or the copy number of different gene families. But what we want to know is this, like when the genes were gained, when the genes were lost, and when they were lost, we need to know whether they were lost because of just random loss, deletion, or because selection against having that gene. And to make things more complicated, this is also going to be affected by duplication. If a gene tends to duplicate very much, it's going to be very difficult to lose it because just you will have many copies of it. So we are trying to reconstruct this history of gene gains and losses, and then we need to disentangle the causes, like we need to disentangle the role of selection for the maintenance of those genes. How do we do that? Well, here, so I put some equations, we did some models. We basically wrote some equations describing this process of gene gain, gene duplication, gene loss, and selection. The equations look something like this. And then what we did was to solve those equations and find the relationships that basically relates the effect of selection with the effect of gene loss. So the idea is that in the end, when we look at a distribution or the evolutionary trajectory of a gene, we just see the effective loss rate. We just see that the gene has been lost from the population, but we don't know if it has been due to chance or due to selection against that gene. So actually what we were able to do was to derive an equation that connects the different causes for effective gene loss in the population. We could also fit the gene copy number distributions to the model. They fit quite well. And with these two ingredients, the fits of the data to the model and this equation here that tells you how the effective parameters relate to the selection, we were able to infer the actual rates of gain, gene loss, and the selection parameter. So skip this. This is what we found. So we did it for different gene categories. I put here some of them, like going from some essential functions like nucleotide metabolism and translation to some non-essential functions like pathogens, prophages, plasmids, transposons, defense, secondary metabolism. Here we have the selection coefficient. So it's positive and high if it's a gene that is very beneficial for survival, and natural selection tends to keep it in the genome. It is red and negative if it is bad for survival, and natural selection tends to remove it. First, we see that, as expected, genetic parasites, selfish genes, are very costly. They have negative contributions, so they are not surviving because they have occasional benefits to the host. They are overall costly. But we also see that antipathogen systems like CRISPR-Cas or other defense systems are also almost as costly as the pathogens themselves. That's telling us that having a prophage in general is not good for the bacteria, because the bacteria is keeping systems that are extremely costly for the sole purpose of fighting those viruses. We also found that functions that a priori could seem beneficial, like carbohydrate metabolism, genes that help you process food in the environment, are overall neutral in the long term. It's here, you see, it's almost zero, which probably means that having those genes is useful. For example, having a gene that allows you to eat, to metabolize fructose is useful if you have fructose in the environment, but if you don't have fructose, it's probably costly. And because the availability of fructose in a long evolutionary times fluctuates, it makes those genes effectively neutral. Okay, so they don't tend to be conserved in the long term, and that's telling us something about how quickly bacteria adapt to the environment by gaining or losing the genes that they need or they don't need anymore. All right. Also, based on the magnitude of this selection coefficient, we can see that those that have values smaller than -1 are mostly controlled by selection. So selection is actively purging them from the genomes, and those that have values greater than -1 are basically controlled by deletion. They are just random loss getting rid of those. So we have seen that these genetic parasites, these selfish genes, are costly, and they tend to be parts by natural selection. How can they persist? Why do we still see them? Why are they so many and they correlate with population size? And there are two possible reasons. Without horizontal gene transfer, there is selection that tends to make less likely to reproduce to those bacteria that have the prophage or the plasmid, so they would disappear from the population. What happens is that I didn't talk about a very important ingredient, which is horizontal gene transfer. These elements tend to be mobile, so they tend to encode the genetic machinery that allows them to jump from one genome to the other. Now they are parasites in the whole sense of it. They are able to infect new genomes. It's the same as a virus or an infection, like, the body fights them, but then the virus or the bacteria jumps to another host, and that's how they can persist. There is another possible explanation, which is that they have this occasional benefit to the host. However, I just showed you before that this is not the case because the overall fitness is negative. So we want to know what is the minimum rate of horizontal gene transfer that a parasite needs to persist in the genome. And then we will check if that's actually the rates that they have in nature to basically close the theory and say that all this is consistent. For that, we develop a model that allows us to basically predict the minimal rate of horizontal gene transfer required for persistence, which is this formula here. Here is graphically plotted. So as a function of the cost of the parasite, if the cost is higher, you need to increase the horizontal transfer rate. Parasites that are very costly, they need to be very good at transmitting themselves to other hosts. Also, we saw, we look at how self-replication within the genome helps survival. And we found that it's not very important. So only for selfish genes that are not very deleterious, it makes sense to proliferate, like is the case for transposons, for example. We compare those predictions with the actual values, and we found that for 4 different classes of selfish elements, toxin, antitoxin, prophages, plasmid, and transposons, the rates of horizontal gene transfer are over this minimum required for survival. So now we know that they are costly and they are mobile and they just survive as actual parasites by propagating to different genomes. It's interesting to point out that not all these 4 kinds of genetic parasites are mobile. Prophages are autonomous. They are able to mobilize in an autonomous way. Same about some plasmids, but toxin, antitoxin, and transposons, they don't have mechanisms to infect other genomes. What they do is to hitchhike with some of the other 2 genetic parasites, and that could be the basis for a number of questions about genomic organization, why toxin, antitoxin tends to be spatially closely located with some other genes like reference systems or transposons. So it's telling you that these non-mobile selfish genes like toxin, antitoxin, and transposons need to actually place themselves near a mobile gene so they can survive. And they have probably evolved to be able to do this. I will skip this part. And yes, I will like to finish with this general picture of what things I've been talking today. So I talk about horizontal gene transfer as a central ingredient in genome evolution. We know that horizontal gene transfer allows the persistence of these genetic parasites, and these genetic parasites, selfish genes, have overall negative effects. These genetic parasites, many of them are mobile, so there is a feedback such that these parasites, by being mobile, are also promoting horizontal gene transfer within the host that they infect. Now, this horizontal gene transfer can also have a positive side because you can also gain useful genes. I didn't talk about this in the talk, but it's easy for you to imagine that you can also get things that help you, for example, fight an antibiotic. And it can also promote sequence divergence. This is a very interesting project that we had 2 years ago that I didn't have time to present, but Basically, what this project told us is that gaining and losing genes accelerates evolution in genes that are constitutive, that are core genes, that are not gained and lost. So there is an interplay between this genome fluidity and the evolution of genes that are markers, like ribosomal genes. And because of that, the combination of these two ingredients leads to speciation. So we have a quite complicated pictures where elements that in principle are negative are promoting gene gain and loss, and indirectly can lead to very important question in biology, which is the origin of species. With this last slide, take-home message, which is that for those of you who like maths or physics, combining comparative genomics and this kind of mathematical model, it's a quite powerful strategy to better understand these principles and trends that underlie genome evolution. Of course, in the case of animals and plants, but also for prokaryotes and viruses. I want to finish thanking my collaborators, the members of Eugene Kunin's group here, where I spent 6 years as a postdoc before moving back to Spain, members of my current group at the CBGP, and also a quite large number of collaborators that I've had during the years and have contributed to the project that I presented today. So thank you very much for your attention, and I will be happy to take any questions.
Thanks a lot, Jaime, for this very interesting talk. Any questions here? We can ask perhaps Izaskun, that I think she controls the online connections. If there are some questions there.
There are no questions so far here.
Okay, I have a question actually. I have a couple of them. The first one, regarding your last transparency, in which you say that even if you've been saying all the time that these selfish genes have only negative effects, that in the long run they also have some positive effects for the general evolution of the biosphere. My question would be, does this potential benefits for the system be in some sense a help for them to survive?
So that was one of the arguments that was given in the past, to say that they are selfish, but they are not. They are also beneficial indirectly, and because of that they survive. We investigated that part. Actually, we investigated what would happen, like if bacteria could curtail all horizontal gene transfer, which it's hard to believe that could happen, but that's a possible scenario. Like hypothetically, imagine that you can actually have a super efficient like system that identifies all foreign DNA and it destroys it and it's never inserted into your genome. And of course, if you do that, you could get rid of genetic parasites, but you would end up losing many genes that you need in the long term. Like you would lose any gene that is not useful today and that could be useful in 100 years, and you would not be able to recover it anymore. And as a result, there will be some sort of Muller's ratchet and the population would degrade. So it's not, I don't look at it so much from the point of the mobile genetic elements. I see from the point of the cell, they need to, like, mobile genetic elements and parasites are the price to pay to be able to escape Muller's ratchet. Population has to keep some level of gene intake to be able to escape Muller's ratchet, and that opens the door for parasites to persist. And then it happens that these parasites also provide horizontal gene transfer. But even if they didn't provide with horizontal gene transfer, if the cells have their own mechanism, and they have, like, for example, transformation, parasites would eventually cheat on that to persist.
Okay. Any questions here?
Yeah, maybe wasn't too clear to me. If you're given a set of genes, how do you actually distinguish the selfish ones from the altruistic ones? What's the algorithm or process?
Yeah, so the way to do it in an agnostic way, I guess you would need to estimate the fitness effect. But to estimate the fitness effect, you first have to cluster them into groups, and there's some statistical limitation if you have very little groups. So for this study, we actually use classifications of genes. So we identify genes that are part of prophages or part of phages, and we put them together as the category prophages, and then we evaluated their fitness cost, same for plasmids, same for transposons. And the thing is that there is, of course, a large number of genes that can't be classified to anything, which many of them can be possibly parasites, which are those in, I don't know, they don't even know here. No. So we had a category that was unknown. That category of unknown was slightly deleterious, and probably in that unknown category, which are things that we can't classify a priori, there will be undiscovered defense systems, selfish genes, and things that are probably false gene predictions and some beneficial things. But it is true, that's a limitation that to start with, we need to classify them functionally, and that we need to have some sort of previous knowledge of what a gene does to do this.
Thank you.
Any other questions?
We have a couple of questions in Zoom.
Wait, there's one question here.
Okay. Thank you. Thanks for the talk. It was very interesting. I have this question. So you've been talking before about the phylogenetic trees. How can we, if we can trace back to what was before, and then about these genetic elements. And some bacteria pathogens I'm thinking about now, they have prophages that are responsible for like having these pathogenicity effects in when they infect humans or other animals. So they have an useful trait for them. My question is related to how can we trace back all these prophages that have this useful benefit for the pathogens in this case, so that we, or if we can like think about how would be the first pathogen that had these islands that were useful for them to infect, if we could do that. It's more a reflection, but more a thought than a question, I think.
So you are, I don't know if I understand, you're asking if there is any way that we could try to reconstruct or make predictions about how the first viral pathogen was like? Or?
Not viral pathogens, but bacteria that infect animals, for example, have usually, I'm thinking about the type 3 secretion system. It's encoded in a genetic island that encodes the system itself, but also virulence genes that cause the infection. So this is something that has been like shared among bacteria, so that they like can infect more animals, plants, and other like eukaryotic cells. But it's encoded in a prophage, so that if we wanted to like build a phylogenetic tree of it, how could we do it? Because they're like with all these horizontal genes transfer and all that. Could it be done? So that's the question.
You can, so if the genes keep enough level of sequence homology, you can make a tree for that gene. If we are talking about an operon that has been transmitted together as an operon for most of its evolutionary history, we can make an evolutionary tree for the operon. If we are talking of an island that has suffered within island horizontal gene transfer, then things get more complicated, because different genes or different components of that island, of the pathogenicity island, are going to have different evolutionary histories. On top of that, you have the problem that if the genes, if you are comparing things that are very divergent, you may be able to know that they are homologous, but they may not be similar enough to make a high quality tree. So even if you may not even be able to make gene trees, as is the case with the viruses for things that are diverse. And that's when you enter into doing like profile alignments and more sophisticated methods. Actually, there is something I didn't show, but in one of these studies, basically it was mostly Yuri Wolf, built a tree of all the RNA polymerases of RNA viruses. So he actually, we published it, there is a tree of all RNA viruses based on the RNA polymerase. But that was super complicated. Like the techniques that he had to use to build this, because they are super divergent, was very difficult. So technically it can be done for single genes, but things get very complicated. And specifically, if you are thinking of a pathogenicity island where the components have different evolutionary histories, the answer is that you can't, because they may have different histories. What you can try to do is to try to trace the origin of the different elements. It has been, for example, done by Kunis group recently with a CRISPR-Cas system, where they try to trace the origin of the different components of CRISPR-Cas system. And some of the components come from transposons, some of the components from other kind of systems. And the evolutionary scenario is that these things that have nothing to do with each other, they happen to appear together in one genome, and then they adapted to work together, and they provided with effects against the host. I don't know if I answered your question. It was a difficult one because, of course, if it was easy, it wouldn't be a question.
I think there was a question in the internet.
Yes, there are a couple of questions. The first one is from Alba Cuecas, and she asks, hi, I doubt that insertion sequences or transposon could not move through transformation to another genome without using plasmids or phage. Which implications would that option have in your theory?
I don't know if I understand the question. So what would be the implications if they could move to transformation or if they couldn't move to transformation?
She says, I doubt that insertion sequences or transposon could not move through transformation to another genome without using plasmids or phages.
Wow, this is double negation. I don't know. So I will answer in both cases. So if they can only move using plasmids or phages, it's telling you that they need to be spatially associated, like linked to spaces, to transposons or phages. That's it. But sorry, to plasmids or phages. Sorry. That's the only way for them to mobilize. If they can mobilize through transformation, then they could be anywhere in the genome. The question is whether the transfer rates that are provided by transformation are enough for them to survive. Possibly they are, because at least based on our results, they are not very costly. So the selection to eliminate them is not very high. So possibly the rates of transformation could be enough for them to survive. So what I can say based on my poor understanding of the question.
She says thanks. And then the next question is from Hugo Bastoya. And then he says, congratulations for the work, Jaime. In your model, did you take into account the possible leakage of biochemical products in bacterial community, for instance, amino acids or products of carbohydrate metabolism?
No, and actually that's a limitation of this project. So all this analysis, like the phylogenomic reconstruction is done taking into account the whole genome, but the quantification of the selection effects and all that is done at the gene family level. So we are not considering interactions between gene families, and we are not considering at this step interaction at the level of the community. That's actually one of my current lines of research, like trying to translate this from the single genome level to the microbial community level. But we are still work in progress.
Okay, thank you. And then, and I have a question, if I may. So I'm a completely outsider, so I may be asking a very silly question. So you showed at the beginning of your talk a method in which you could classify the different viruses into the different families. And then you found that there was a one-to-one correlation between your classification and also the one provided the taxonomic classification you mentioned. I was wondering whether, so that one-to-one correlation, so you do it because you know all the genes within for a virus. So that's how you make the one-to-one comparison. So you need to have the full information, genetic information from a virus. Is that correct?
Okay, let's see if I understand. This correlation appears as a result of the clustering of the network. So what does it mean? That when we generate the network, we don't know the taxonomy, or we don't give it any kind of taxonomy. When we use this community detection algorithm, we don't give it any information about the taxonomy. So it's all completely taxonomy agnostic. We build the network, we find the modules, and then we compare. We wanted to see if these modules have something to do with taxonomy or have nothing to do with it. And then we found that there is actually a correspondence with the taxonomy that is provided by biologists when they study the genomes. So in that sense, it's independent things. Now, you mentioned something about how having complete genome, having all the genes, how important it is. The fact is that it is important that the genomes are basically that there are no biases in the content of the genomes. If they are incomplete and you are missing, especially in the case of RNA viruses that tend to have small genomes and just a few core genes, or say like genes with phylogenetic value or classification value, then if your genome is incomplete and you are missing that gene, then you are screwed, because you are. That's not the case in double-stranded DNA viruses and viruses with larger genomes, but in RNA viruses, yes, if your genome is missing an important gene, things may not work. However, that doesn't mean that the algorithm is using all the genes. I didn't mention that you have genes that have more power for this kind of community assignments, and genes that are more like patchy distributed, and you can even remove them from the network and nothing changes. So it depends on which genes are missing, and that's something that once you have a reference network and you have doing it, it's easy to see. Like you can filter that network, identify which are important, let's say holding together the network and the modules, and you can just focus on studying those. You can forget about the rest. But all this idea of like identifying the important genes, it's agnostic of the taxonomy. You don't need to know that, and that's the key of the method.
So then in principle, so thinking about the phylogenetic tree, I was wondering whether your method would have a predictive power to actually, by selecting or by knowing all these key genes, whether you could fill up all the phylogenetic tree by knowing the key information. Maybe we can discuss it offline.
Yeah, maybe. I mean, I don't know if you have my email or.
Yeah, I have your email. Don't worry. This is Itaskun, so I'll send you an email, and then maybe we can have a
yeah, sure.
Thank you.
Welcome.
Okay, last questions here. Then let's thank again the Jaime Iranzo, our speaker today. Thank you very much for being here and for this fantastic talk, and see you next week. Bye-bye.